Uh, Good morning. I would like to extend a warm welcome to all the staff of ICAR-CAFRI, officers in charge, and the team members from our Acre Pond Agroforest Centers, and others who have joined for this workshop on importance of plant variety conservation and the implication of PPV&FRA. To this learned audience, it will be not needed to explain the significance of plants per se in our food web, wherein our entire food system or our life depends on plants. So what is the need to conserve the diversity, and what are the means that has been implemented? As simple means will be PPV&FRA. So all these things are well known to you. And today's workshop will be like what I say, two objectives. One, we have a guest speaker, Dr. K. Rajarajan sir, our own scientist. He'll be dealing about the implications of PPV&FRA and the significance of plant germplasm conservation. And then followed by, we'll have an open-end discussion about what are the steps that has been taken up by ICAR-CAFRI regarding germplasm registration, and what is the steps that we can go ahead. So this will be the nutshell of our workshop. So now I invite our director, Dr. A. Arunachalam sir, to give his opening remarks. Sir, over to you, sir.
Okay, good morning, friends, and Happy New Year to each one of you who have joined online. And this is a very exclusive and interesting workshop that is being organized by CAFRI, just to enable you to have a glimpse of what we are doing with our germplasms, bioresources. And if you have anything interesting, we must also record and register them as a IPR source for the benefit of the humankind for further exploitations and utilization. Now, it is a matter of fact that we all know the importance of germplasms, particularly from the viewpoint of crop improvement or tree improvement. And as we speak, we are also well aware of this Protection of Plant Varieties and Farmers' Rights Authority. And it doesn't mean that it holds the rights of only the farmers, but also the associated people like you and me, who are also exploring and developing, screening and identifying the best land races, testing them, characterizing them, and then finally coming out with its prospects. That we call it as bioprospecting. So in order to take this forward, we thought we will have this sensitization come hands-on workshop, whereby we all will be given to know how to go about if you have some important germplasm in hand, which you would like to take on for registration purposes. So a very simple and pro forma has been developed. And this is not only for the scientists of CAFRI, but also all the people who are involved in AICRP agroforestry network, and also others who are interested in the whole process of biological conservation. So I'm sure this workshop where Dr. Rajarajan is going to be giving you some background information, and followed by the hands-on inputs, how to go about filling up the forms, and whom to contact, whom to send. All those information will be given to you. So have some inputs, and if you have anything more to add, that can be discussed in the end. So with this note, I wish all of you a great workshop ahead. Thank you so much.
Sir, thank you, sir. Thank you for your opening remarks. Now it's time to start our technical session. I would like to invite our own scientist, senior scientist, Dr. K. Rajarajan, genetics and plant breeding, to take up the floor and share his view presentation. Dr. Rajarajan, sir, over to you, sir.
Yes. Respected director, sir, and organizing team, I would like to thank for this opportunity. So today we are going to see about the overall activities of PPV&FRA and the importance of plant variety protection. Am I audible, sir?
Sir, you are very audible, sir. Screen is visible, sir.
Thank you. So before start the session, I would like to, all we know that the germplasms and related terminologies, but still I would like to have a kind of refreshment before entering to this subject. So what is germplasm? Germplasm is a crop species or wild species should have all the total hereditary material, all the available alleles for various characters or the desirable traits. Likewise, land races. Land races are the primitive varieties evolved through non-systematic plant breeding approaches. And of course, land races, many land races evolved thousands years or more than thousands years ago without a systematic plant breeding approaches. Of course, these land races evolved as well, kind of natural selection as well as man-made interference. And coming to the breeding lines are the group of individuals or populations developed by systematic plant breeding approaches. For these breeding lines includes near isogenic lines and many mapping populations like F2 populations and double haploid lines. All these will comes under breeding lines. And coming to the genetic stocks, these genetic stocks generally, these lines carrying gene mutations, either spontaneously or induced. So from a normal line or natural one, due to some spontaneous mutation or induced mutation, the another version of that line used to called as genetic stocks. And coming to the wild forms are the crops directly evolved from its wild relatives or wild species, are called as wild forms. And wild relatives also, during the many crops, during the evolutionary aspect, along with the crop of interest, many wild relatives has contributed to the species development. So these all the species are called as wild relatives. And absolute varieties. Absolute varieties are, once upon a time, these varieties are as a ruling variety, but not now. And many examples are there for absolute varieties. For example, K series of wheat varieties, and so many varieties are there. And coming to the varieties are the ruling varieties, which is systematically developed by plant breeding approaches. So these are some of the important terminologies we should know while handling this topic. And coming to the germplasm conservation, germplasms are the valuable genetic resources, and this needs to be conserved in order to maintain its genetic basis for various crop improvement or plant breeding activities. This allows the production of selection of varieties with desirable characters. And these germplasms later we can utilize and make improvement for our food, fuel, and health sector. And of course, these germplasms, as I told earlier, this germplasm may be any cultivated species, or it may be a wild relative, or it may be a land races, or it may be a wild farm. So these germplasms are ultimately utilized in plant breeding for crop improvement. So if you see that the wild plant genetic resources, if you see all the wild relative species for those which are involved in food crops as well as health sector, medicinal plants, as well as for wood and timber. Also, these genetic resources also used in food and agriculture. If you see that in non-food utilization, mostly ornamental species, fuel, paper industries for wood related, these species will come under. And coming to the method of germplasm conservation, the purpose of germplasm conservation is to protect the genetic diversity from genetic erosion. And of course, when we will deal about the plant breeding, and due to various man-made activity and anthropogenic activity, the genetic variability is getting narrowed down. And in order to avoid that, the germplasm conservation is important. And germplasm conservation is primarily, we can classify into two major classes, like ex situ conservation and in situ conservation. And in situ conservation means where the natural habitats of these plant species are there. In the natural habitat itself, the plant species have to be conserved. These criteria used to called as in situ germplasm conservation. Generally, these used to come as biosphere reserve, national parks, and where the primary origin of these species are, those will come under this class. And coming to ex situ conservation, these are away from the in situ conservation areas, like field establishment of field gene bank, seed orchard, and in vitro conservation, like cryopreservation, DNA gene bank, or shoot tip culture. All these classes of conservation will come under ex situ conservation. Why germplasms have to be conserved? Germplasms are the valuable genetic reservoirs resource. So these often house more genes and valuable gene combinations for various crop improvement, and to depending upon the human needs from time to time, these germplasms can serve the valuable gene combinations for improves the productivity, it increases the resistance, it increases the adaptability to various climatic factors. So this is the reason we have to conserve the germplasm. Also, the germplasms are the prerequisite for any crop improvement in program. So these germplasms, if you see the conservation steps, if you see first the acquisition of germplasm, it means any exchange, exploration, collection, these activities will come. And then we have to go for conservation. This conservation includes active conservation, working collection, and base collections. And depending upon suppose base collections are there, means we have to store these collections for 3 years, up to 3 years, and under less than 5 degrees Celsius. And of course, with the mostly orthodox seeds, we can store with 5% moisture content. Likewise, active collection and working collection. Working collections are the collections we more often we used to maintain in the field for various crop improvement programs. And coming to the management, multiplication, regeneration, and testing of frequently, we have to do these practices to manage the germplasms. And after that, we have to go for characterization. Characterization includes evaluation of those germplasm, documentation, and distribution afterwards. So once the characterization comes, then this can be better utilized by various plant breeding methodologies to have a product like new varieties, crop species, and also this can serve on-farm diversity as well. And coming to the status of global germplasm conservation, as of now, a total of 5.43 million accessions are stored or conserved in across the countries in the globe up to 2019, in which 5.8% are are conserved in field collections. And the USDA ARS National Plant Germplasm System holds majority of the accessions and covering about 15,970 plants. And of course, this USDA ARS, the system holds 73% of collections of the total as of now. And apart from that, 7 CGIAR gene banks across the world, country around some 44 countries, they are having their centers, in which ICAR platform alone have 11,000 accessions of tree species and nuts. And this is the scenario of accessions from 2016 to 2019 onwards. And coming to plant variety. So, plant variety is the result from the selection and deliberate control of plant species by various plant breeding means. And this involves systematic plant breeding approaches, right from selection, introduction, hybridization, and then it's a controlled crossing, and then the variety by evaluation of various locations for yield and yield trials and various targeted traits. And then finally, this variety has been evolved. And the breeding new varieties requires skill, time, money, and material resources. It requires a lot of skill to develop need-based variety from time to time, of course. And these activities enhance. Ultimately, the aim of the development of new variety is to enhance the growth of agriculture. And by the way, it boosts several emerging economics in both developing and developed countries. Ultimately, it enhances the production and productivity of the concerned crop species. And why we have to do all this thing is ultimately to enhance the country's agricultural growth. And therefore, this variety also, in the same time, this variety also safeguard the interest and further development of plant varieties by plant breeders. And this variety need to be protected through intellectual property law. So the intellectual property law here, we can say it as protection of plant varieties and farmers' right. Okay. So the aim of in the earlier slide, I told that the plant varieties needs to be protected because of we have to, those varieties will have the valuable forms for increase the production and productivity. In the same time, the variety, the rights of farmers and the plant breeders to encourage development of new varieties from time to time. So the government of India enacted the Protection of Plant Varieties and Farmers' Right Act 2001, adopting sui generis system. Of course, the act follows UPAV, International Union for Protecting New Plant Varieties, 1978 and 1991 as well. And also it accepts the obligations from TRIPS, trade-related intellectual property rights. And the legislation recognizes the contribution, but our PPV&FRA Act 2001 recognizes the contribution of both commercial plant breeders and farmers in plant breeding activity as per the TRIPS agreement. And to effectively implement this act, Department of Agriculture and Cooperation, Ministry of Agriculture and Farmers Welfare established Protection of Plant Varieties and Farmers' Right Authority on 11th November 2004. So, under this PPV&FRA, the registration of plant varieties are the major emphasis. So, what are all the varieties can be protected under this? It's a new variety and essentially derived variety and extant variety and farmer's variety. New variety means expression of a particular character of interest from a given genotype. This is called new variety. And essentially derived variety is an expression of essential character from an initial variety, the variety which is already been developed. And from that variety, it may be any essential characters. It may be related with resistance to any pest or disease, or it may be a high yielding QTLs, or it may be. And that particular expression of that particular trait from an initial variety is called essentially derived variety. And external variety, the external variety, it may be a farmer variety or variety about a common knowledge or which is available in the public domain. So this is called external variety. And farmers variety also can be protected under this. Traditionally, the farmers long been cultivated traditionally and evolved farmers field, or it may be a wild relative or land races. So these varieties also can protected under this act by PPV&FRA. And for each, these crops are depending upon the species, there is a duration of protection. And most of the crops, the duration of the period is 15 years, the protection period. And for us, trees and vines, it is up to 18 years. And the external variety, which is already notified, is 15 years. And for registration also, it's for farmers variety, they are exempted from the registration fee, and others, they have a registration fee for external variety, essentially derived variety and new variety for individual and institutional and commercially. Depending upon that, the fee structure is getting changed. And apart from that, in this PPV&FRA, there are 3 rights are there. One is plant breeders' right, another one is farmers' rights, another one is researchers' right. Okay, suppose a plant breeder is developed a variety, who developed the variety, and he ultimately he gone for protection of that variety. Once the variety is protected, the certificate is issued. Without the concerned breeder, the variety cannot be reproduced or cannot sell in market, or it cannot be exchanged in anywhere. And likewise, the farmers also have its right that the farmers variety can also protected in such a way. And farmers also have this kind of rights that without his own consent, the variety cannot be reproduced or marketed, or it may not be exchanged, and it requires a consent from the farmer. And however, for the researchers, these exemptions, there are some exemptions that they can use these materials, these protected varieties under this act for only research purpose. And by using these protected varieties, a new variety can be developed, and repeated use of this requires again a permission or a authorization from the concerned breeder. It is required. So this is about the registration of plant varieties. And of course, all the plant varieties which are registered are must to go DUS guidelines, which are developed by PPV&FRA again. And this is the organization of PPV&FRA. This is chaired by a chairperson, and then a registrar general, and then 15 members are there, in which 8 ex officio members, and 3 from state government SAU, and 1 from each tribal and women's association, and from farmers group. This is the organization of PPV&FRA. And objectives of this PPV&FRA, main objective is to establish an effective system for protection of plant varieties. And then rights of farmers and plant breeders to encourage further to develop a new varieties. And this is the main objective. And then to recognize and protect the rights of farmers in respect of their contribution made during the conservation or improving and making any available plant genetic resources for the development of new varieties. And also, ultimately accelerate the agricultural development in the country by protect plant breeders' right. the way it stimulates the investment for research and development in both and public and private sector, and also simultaneously facilitate the growth of seed industry, to facilitate the growth of seed industry in the country, which will ensure the availability of high quality seeds and planting material to the farmers. Coming to the functions of PPV&FRA, the main function of PPV&FRA is to register the new plant varieties, EDV and extant varieties, and developing various dust guidelines for new plant species. Dust is nothing but distinctiveness, uniformity, and stability. And these are any variety should be protected under this act. It must have this dust characteristic feature according to the PPV&FRA dust centers. As of now, they have more than 122 dust centers for different crop species across the country. And developing characterization and documentation of varieties registered, and compulsory cataloging of the varieties registered for all the plant species, and documentation, indexing, cataloging of farmers' varieties as well, and recognizing and rewarding the farmers' community, particularly tribal and rural community, engaging conservation, improvement of plant genetic resources for various economic plants. And last, maintenance of national register of plant varieties and national gene bank. So these are the main functions of PPV&FRA. And as I told earlier, the registration and requires it must fulfill the dust characteristics. And the central government issues us notification in official gazette specifying the genera and species for the purpose of registration of varieties as well. And the registration must go through, if suppose a farmer is registering this variety, he can directly may register, or he can go with any agent or through any institutional he can go. Likewise, any individual who is associated, who is the sole responsibility for developing the variety, can go for registration. So there are two awards and recognitions for the farmers as well under this PPV&FRA. One is Plant Genome Savior Community Award. This award is given for the farming community, a group of farmers or a community of farmers. So in which each year 5 awards they used to give with worth of 10 lakh rupee each award. And the second one is Plant Genome Savior Farmer Award and recognition, in which each year 10 rewards with cash price of 1,50,000 and with citation and memento, as well as 20 recognition with 1 lakh cash prize with citation and memento, also to encourage the farmers in the field of conservation of genetic resources and to develop a new variety. To encourage them, these awards have been issued from in each year. And so far, the PPV&FRA issued 5,100 certificates, and it started issuing the first. It started receiving the applications from 2007 onwards, and now 5,100 certificates issued. In which, if you see that the cereals having highest number of certificates, that is 3,394, and trees also have 8 certificates issued for trees. And then coming to the different varieties, if you see that farmers variety, 2025 farmers variety they have issued so far, and essentially derivative 14 varieties, like new varieties 937, and certificates so far issued. And these certificates are issued by the registrar or the member secretary of this PPV&FRA used to issue. For more information, because of this time constraint, I have narrated only the major functions of this PPV&FRA. For more information, you may go through the plantauthority.gov.in. And coming to the Institute Germplasm Identification Committee, ICAR-CAFRI has its own Institute Germplasm Identification Committee, which is chaired by the program leader Tree Improvement. and it is the committee functioned through member secretary, and it has own members of scientists of this institute, and it has a provision for adoption of new need-based tree crop specialist with reference to material under consideration. For though depending upon the material consideration, a need-based specialist will be looking after to evaluate all the plant germplasm and genetic resources related activities of this institute will be handled by this IGIC committee. So this I would like to give the guidelines and some the informations to be followed for their germplasm activities. So the eligibility criteria for germplasm registration at MPP&FRA, it must go through this IGIC. The germplasm genetic stock, allied material of field crops, horticultural and other economically useful, which should be unique, uniform and stable, and has potential attributes of academic, or it may be a scientific, or it may be a commercial value. And exotic germplasm that is imported from foreign countries can be identified and registered for trait other than those already published or registered. Similarly, selections made from exotic germplasm can also be recommended for registration. Selection for unique traits from land races other than the land races is already known for may be identified and recommended. These criteria are eligible for germplasm registration. And of course, the one who sends for germplasm consideration or for registration, they must attach supportive files for the same, these are different depending upon the traits, like quantitative trait or it's a qualitative traits like that. Suppose performance, yield contributing traits, adaptation traits, qualitative traits, data for at least 4 environments, that is location and year combination, under All India Coordinator Research Project trial test for qualitative traits like flower size, shape, and other leaf venation. Data, 2 different environments is required, duly supported by the documents. The supported documents may be, the data related to the qualitative traits, clear photographs. All these are required. So for resistant tolerance to biotic and abiotic stress, data should be obtained for at least 4 environments under established hotspot locations or under artificial screening conditions. For biotic stress in annual biennial crops, inheritance of the trait should be worked out. How much the inheritance is there, the resistant gene or the essential trait is inherited from one generation to another, that should be ensured. And for all this, the supportive biochemical evaluation is required at least 4 environments, and these biochemical indices or evaluation also needs to be submitted at the time of evaluation or consideration for registration. And the proposed genetic stock germplasm should also be evaluated along with the registered genetic stock germplasm. This is mainly to compare the new genetic stock performance in respect with the existing one, in order to show its uniqueness or its superiority. And there are some germplasm ineligible for registration. Under this, the germplasm or genetics are without accompanying a documentary evidence for the claim made in the application. As I told earlier, the documentation is very, very important, which mainly supports the uniqueness or the superiority of the germplasm. Also, the germplasm or genetic stock does not contain complete passport data and the uniqueness of the germplasm. Each germplasm must have the complete set of passport data. And without passport data, the germplasm may not be considered. And exotic material per se with no evidence of human intervention in its improvement. And varieties, hybrids of common knowledge or selection from traditional farmers' varieties. This cannot be considered. And germplasm which involves any technology which is injurious to life or health, human being, animals or plants, or like terminator gene technology like that, this technology involves, then this material won't be considered. And material for which any form of protection has been sought elsewhere, also not considered under this criteria. And screening of application and their consideration by the IGAC. Once the application is submitted from the applicant, then the IGAC will look into it and screen the application as per the guidelines fixed by the committee. The member secretary IGAC will screen the proposal submitted to the prescribed proforma. Then each proposal will be reviewed by the IGAC, and this review will be carried out by specialist or expertise depending upon the need or the plant species are submitted. And then it finally placed for the approval of the director or the project coordinator. And then after initial screening, incomplete application would be advised for appropriate revision, or if it may be rejected on the technical ground. And the application in which the validation of the data is considered necessary, the applicant would be required to produce a validation report from an appropriate institute as advised by the member secretary. The data is considered, validation of data is highly necessary because with the validation of the data, the IGAC will mainly consider. And coming to the germplasm information in CAFRI, we have germplasms of Azadirachta indica, Pungamia pinnata, and Albaja siso. These are the germplasm we have at our CAFRI center, and these germplasms are maintained in field gene banks. And of course, this germplasm also being evaluated for various end users, like for neem, we are evaluating for high kernel yield and oil content. And Pungamia also, we are evaluating the germplasm for oil content and biodiesel property. And Leucona leucocephala, we have the accessions, and these accessions are being evaluated for various wood properties like fuel wood property and pulp wood property. So these are all the germplasm information which is available at CAFRI, some of the information. And thank you. And now let us move for how to fill the application for germplasm registration or the germplasm identification. It must go through the germplasm identification committee. And how to fill, get to the IC number for to take the passport data.
Sir, do you want to unshare the screen, sir? Oh, okay, thank you.
Okay, so this is the application for Institute Germplasm Identification Committee. The one who is working in germplasm related activities, either in the institute or in the ICAR centers, ICAR on agroforestry, in different centers who is dealing with the germplasm, or they wanted to register the germplasm at NPPGR, or they want to, they need to have a IC number or EC, exotic number collections from NPPGR. They must go through this application and they have to submit their findings or the materials to ICAR-CAFRI Johnson. So I will show how to fill this application. This application have 3 different annexures first, and this is the main application. And these are the annexures. So first, if you see that application status, whether application status means whether the application N or R will be there. Suppose this is a new application, means we have to write it as N. Suppose it is an old application, or it is already submitted, it requires a revision. It means we have to denote R. And crop name, the crop name, it must very clearly we have to write the crop name in common name. Suppose it is rice or it is wheat, we have to write it as rice or wheat. Then botanical name of the concerned crop species. Suppose if it is rice, we have to write it as Oryza sativa. It is neem Azadirachta indica, we have to write it here. And then crop group. The crop group means we have different crop groups are there. Say for example, if you see in the annexures, these crop groups are available. CL means cereal and pseudo cereal PC. Suppose FG forage, FT fruits, F for forestry. Suppose it is a commercial crop like any commercial crop, if you wanted to register it, you can denote accordingly CC. So based on this annexure, you can fill this application. And coming to the biological status of the material to be registered, whether suppose when we register the germplasm or any genetic resources or material, it may have different biological status. Either it may be a germplasm collection, or it may be a genetic stock, or it may be a recombinant line, or it may be a near isogenic line, or it may be a mutant. So accordingly, there are codes available here. Suppose if it is a germplasm collection, denote it as GP. If it is a recombinant line, denote it as RE. Likewise, you have to fill this application. Then identity, and if that identity you have to give, then where it is identified. Then criteria for registration. You have to fill it as whether suppose you have identified one unique character, maybe in for qualitative trait, fruit color is different from the existing one. That maybe you have to denote here. Suppose apart of other than a fruit color, as also you have identified some anatomical feature, like suppose you have nectar cavity, 5 nectar cavity, suppose 4 nectar cavity, that you have to fill it over here. Suppose if it shows improved kind of quantitative traits, that also you can give. So these are all the unique features, maximum 3 given. At least when you are submitting minimum, at least you have to submit 1 unique criteria, which has to be comparative with the existing one. Nature of genetic material. So nature of genetic material is nothing but it may be a seed, or it may be a tuber, or it is a vegetative cuttings, or whole plant. Like it depending upon, you have to give the code. So if you see here, nature of genetic material is, suppose your nature of genetic material is seed, you have to write it as SD. Suppose you are providing a vegetative cuttings, then you have to write it as a VP. Likewise, you have to fill this application. And then quantity of deposited. Actually, when you submit a material for registration or consideration, you have to submit a quantity of seed material, or the material you have to provide. Suppose large size of seeds are there, 100 number of seeds you have to give. Suppose it is a self-pollinated one, or it is a cross-pollinated one, depending upon that 1,000 seeds or 2,000 seeds you have to provide. Also, while along with the application, you have to submit this also. Then coming to this values referred by the applicant, whether it is SC, whether the material which you are submitting, whether it is a scientific interest, or it is a commercial interest, or it is a academic interest. Suppose the material is scientific interest, it means suppose your material is dealing with any thing which is anatomical, or it is taxonomical related. Those things will come in the form of scientific importance. Suppose your material is having a commercial value, then it comes under commercial, that is CM. In that code, it must come. Suppose if it is related with its something like its evolution or its biological thing, then this will comes under academic interest. It is ESC. Then basis of eligibility. So basis of eligibility means the material which you are already submitting here or interested to submit must to be published somewhere, either in the form of AR means this is an institute annual report. PR means peer-reviewed journal. Then it is CT, All India Coordinated Trials data. And then any other report on the material which your interest is must to be published in any of this form is eligible to have considered further. has it's been registered or protected anywhere, you have to tick it accordingly. Suppose your the germplasm is already protected somewhere, or it is registered and from which you are developed, that you must accordingly write it, whether it is registered or protected, means yes or no, you have to write. And it is very mandatory that when you submitting all this material, 1-page manuscript is mandatory along with your material submission. That manuscript should contain about a small introduction about the species or the material which you are submitting, and about your unique characters, what you have identified, and then the current cultivational practices and the implications of your unique trait with the current practices. And apart from that, you have to submit the clear photographs of your materials which you identified or which you like to interested in consideration. So that must accomplish 1-page manuscript. And this is for the committee purpose. And once your application is received, the application number will be assigned. Then the date of application, whether the application we have submitted is a new one or revised one accordingly, this will be filled by the committee. And then once this is for the official purpose, once everything is cleared from here, we will assign a reference number. And then if suppose again finally if the committee is satisfied with the validation test which is conducted by the members, or if it is sometimes the committee requires a validation again, then in that case the material once again fully validated, and then the application will be forwarded for the approval. And this is particulars of the scientist or person who are farmers or farmer community who is associated with the material development, their full name, and then their designation, their address, email, if it is available for farmers variety. We must fill all these data. Suppose for germplasm registration purpose, you may have already, if you may have the germplasm passport information. If so, you have to mention the indigenous collection or exotic collection number. You have to mention any other identities which is related to the germplasm and source of collection, whether it is institutional collection or it is from the farmer's field, or where from the source of origin, and that you have to mention. And the place of origin, provenance, and district, and this geographical location must be filled out over here. And if also it is required that genes, if any identified, any functional genes or any genes or QTLs which are identified also needs to be filled it over here. And during the process, suppose you may be taken this variety from the farmer's field or a farming community, also you have to give the recognition, and you have to give, suppose the farmer or community made any contribution, that also must be filled it over here. And suppose for the breeding method, if you see the genetic stock, when you are developing a genetic stock, it is mandatory, must give the pedigree information of the genetic stock, where from it evolved and which material it is evolved, what is the parentage, what are all the pedigree history, that must be given. And breeding method, through which breeding method you have evolved this genetic stock. There are different breeding methods are there in the annexure. If you see introduction and selection means IN, pedigree selection PS. Suppose it is a recurrent selection, it is RS. Suppose backcross derived genetic stock, you have to give as BC. So depending upon the pedigree and breeding method you use, you must fill this column. And then salient characters are trait of description. You have to mention your trait description and what way the trait of interest going to be registered, and what way it is being sent for consideration, and what way it is unique from other. So these information you have to list as per this. And of course, you have to mention also the trait or the character which is identified from the genetic stock or germplasm. What is the usefulness of that identified material? The usefulness, as I told earlier, it may be a commercial use, it may be academic use, it may be a kind of, it's a scientific way, it may be useful. So that the usefulness of the germplasm or genetic stock must be mentioned over here. And you have to mention about also, you have to mention about the production, seed production capacity or the seed production status you have to mention, and where you are producing the seed production that needs to be mentioned, and quantity of seed available, and how much is the quantity of seed currently available, and the seed viability percentage and germinability. All the details must be accompanied with this application. And apart from all other, any additional information you would like to give, and along with this information, you may provide it over additional information remarks. This is once if you filled all this application, and this, the undertaking must be signed by the depositor or the proposer or their associates, whoever is involved along with this activity, all must be signed in this application. And then this application needs to be submitted along with the appropriate genetic material or interest to their committee. And coming to this is for the proforma for passport information for getting IC number 10B, and this is also need to go through the IGIC committee. If you, suppose you have 2-3 species, you have identified different species, you have to mention here one by one. And national identity, you have to mention where from it is, and that collector number, donor institute. Suppose it may be collected from any institute, then you have to mention donor other identity. It may be from any farmer field, or it may be from any other source. You have to mention here. And like the crop name, you have to mention, and the common name, the taxonomic code you have to mention. Then pedigree, suppose it's a genetic stock, again I am saying the pedigree information has to be filled, and source like institute, farmer field, or it is market or any non-governmental organization. This has to be filled. Country of origin, location, longitude, latitude, this GPS information has to be filled. And if any other remarks, this has to be filled. And to fill this, there are some legends are available in below the table. To follow this, you may fill this application. And then checklist for screening of applications. And then the member secretary, ICAR-CAFRI at CAFRI will screen this application based on this information. It is whether this is a new application or revised one, or it is similar like earlier one. Like these are all the criteria they will consider for screening while screening the application. And checklist for reviewer and expert for recommendation. This is the recommendation made by the expert or reviewer who's going to review the material. Importance of trait. Though the applicant has given the importance or the usefulness of the trait, it is also a duty of reviewer to validate such a usefulness or importance of the trait coming under the same criteria or not. That reviewer will follow this, and he will see whether the data as per the guidelines the applicant has submitted or not, and then validation test is further is required or not. This has to be decided by the reviewer or the expert who is going to review this. And finally, they will recommend the registration. Once the recommendation is made, the director will approve this, and followed by this will take it into the further action. So this is about the IGFT committee activity and how the application to fill it. Thank you very much for this opportunity.
We are reaching about the last stage of this workshop. Now I'd like to invite our Dr. Subin Thomas, SRF of our ITMU unit, to give a vote of thanks.
A very good morning to all. On the behalf of ITMU, I am very glad to be here to express my vote of thanks. A special thankful to our director, sir, Dr. A.R. Alansalam, sir, who allow us to organize this workshop on importance of plant varieties conservation and its implication of PPV and FRA, and its valuable thoughts has truly inspired us a lot, sir. I am thankful to our speaker, Dr. K. Rajarajan, sir, senior scientist, genetics and plant breeding, who accept our invitation and really enlightened with your knowledge, and the whole section which you covered, sir. Really, it's inspired us a lot. Thank you, sir. And thankful to our Dr. A.K. Handa, sir, Principal Scientist and Head of Office for his opening discussion in this workshop. Thank you, sir. And I'm also thankful to our Suresh Raman, sir, who organized this workshop and his effort on that. I'm also thankful to express my heartfelt gratitude to all who participate this workshop through online mode and participate in this discussion also. Thank you. Thank you so much.
Thank you.
Thank you.
So thank you, everyone. Thanks for joining. And we are here. We'll be in ready to answer any of your queries. Please do write to us email. ACRIP centers, you are you can contact us on our ACRIP official website. And with this, we'll wrap up this today's workshop. Thank you once again for joining today. Thank you so much.